Hello everyone, my name is Noah Decker, and today I'm going to be talking about micro-rheology of complex fluids. So first we need to know what rheology and micro-rheology are. Well, rheology is essentially the study of the flow and deformation of matter, where micro-rheology is studying rheology, but at a much smaller scale. A key element that is associated with rheology is viscosity, which is the resistance to fluid flow. You can think of this as a kind of friction for fluids. Next, what is a complex fluid? Well, simple fluids like water or methanol would behave like you might expect a liquid to behave, where complex fluids, on the other hand, may exhibit solid-like properties as well as liquid ones. For example, the complex fluids that we studied involved colloidal suspensions. So we have these liquid emulsions where one substance is dispersed in another, and these colloids can form crystalline structures, which is something we might typically observe in something like a metal. Now, this research is unique because we can measure the viscosity of complex mixtures all under a microscope. And it is useful because we can begin to understand the material properties of these fluids at the microscale, which is something we might not be able to do otherwise at a larger scale. Now that we know what a complex fluid is, let's explore each of the components of our complex fluid. So here we have a video that captured fluorescence imaging under constant temperature heat. The bright particles you see are our fluorescent tracking particles, and the dark region is the water. Now you might notice that these particles are moving randomly. That is because they are undergoing random motion as a result of being bombarded by water molecules. We call this Brownian motion. We are actually able to track and calculate this motion, and I'm going to show you how we did it. So how do we track Brownian motion under a microscope? Well, we place our medium with our tracking beads under the microscope, and we take images at points in time. Next, we use software that is available to us to track the trajectory of our particle in the frame of our microscope images. Now, I know that there's a lot of interest today in AI and machine learning, deep learning, to train large data sets, many particles at once. We are looking at particles that will stick to each other, that will go out of frame during the time that we're in. Once we track the trajectory of our particles, we can calculate the displacement of their position at each point in time. We can square each of these values to have a positive displacement and account for movement in any direction. By doing so, we can calculate the mean square displacement for all of our particles at each temperature. Here, we needed controls for each of the components of our complex fluid so we could compare our values to something. Now this graph shows our mean square displacement for water, where we held our temperature constant every 5 degrees Celsius. Once we have our mean square displacement, we can relate this to our diffusion through the medium. This tells us that our diffusion constant is equal to our MSD, or our mean squared displacement, divided by 2 times the number of dimensions, in this case we have 2, our x and our y position, times the time. Now this means that we can take the slope of our MSD versus time graph, divide by 4, and obtain the diffusion of the medium at each temperature. Now here our particles have slightly different sizes, which is going to influence the range of our diffusion. Therefore, we have a range of values that's considered to be acceptable for a medium of water. We'll be able to see this relationship in an equation I will show later. All of our values fall within this range, so we are good to go. Once we have our diffusion, we can relate this to viscosity using the Stokes-Einstein equation. This tells us that viscosity is inversely proportional to diffusion, and is influenced by the thermal energy of our system, or the Boltzmann constant K times our temperature T that you see there. From this, we can calculate our experimental viscosity. We can also calculate our theoretical viscosity of water using that equation that you see there for theoretical viscosity at any temperature. And this equation resembles some sort of Antoine's equation you might see in thermodynamics. Now, using this equation, we can also go backwards to obtain a theoretical diffusion, which gives us that range that we saw in the previous slide. Lastly, we can measure a relative viscosity as a comparison between the viscosity of our medium and the viscosity of water. In this case, our medium is water, so the viscosity of water divided by the viscosity of water should give us 1. Our experimental values are in agreement with that. Now we can move on to the second component of our complex fluid, which are colloids. Our colloids are basically oil droplets that are dispersed in water. These droplets are usually monodisperse, which means that they are uniform in size. In our medium, now we have water, we have the tracking particles you see on the left, and these colloidal droplets. Now, these two videos that you see here are of the same medium in the same frame. The part on the left is showing the fluorescent imaging of this mixture, while the image on the right is showing the bright field image of the medium. If these colloids were not present, our tracking particles would be allowed to move freely throughout the medium, but the presence of these colloids is hindering their movement slightly, which that is going to affect our MSD, our diffusion, and the viscosity of our medium. Finally, we have the last component of our complex fluid, polymers. Now, these polymers will aggregate with an increase in temperature. So you can see there that we have these droplets that are growing in size. We have already increased the heat in this experiment, and now we're holding this mixture at a constant temperature. These polymers can enter colloids, which is going to cause them to swell, which is something we're going to see in a little bit. Now we can look at the relative viscosity of a more complex media. So now we have introduced polymers and colloids. So for fluids containing polymers, we can see this thickening effect occurring as the temperature increases. So as we're heating, the viscosity is increasing and our fluid is slowing down. For the colloids, we're starting at a high viscosity, but we see the viscosity decrease with temperature. So now we have proven that we can extract the viscosity of different media all from a microscope. Now that we've looked at all the parts of our complex fluid, it is time to put them all together and look at our complex fluid itself. Now, as a reminder, this medium contains fluorescent particles, which can be seen on the left, the water, which is in the background behind all these droplets, the colloids themselves, and the polymers. So now we can see that polymers have caused these colloids to swell, and they're much bigger in size than they were before. Now, if we look closely, we can also see these grain boundaries throughout the images, where the droplets are becoming misaligned. Now, this is because there is a crystal that is being formed in this complex fluid. When we look at the diffusion of our complex fluid, we need to define something that we refer to as the phi number. Now, this phi is a ratio between the volume fraction of polymer to the volume fraction of droplets in our medium. We can see that as our phi increases, the diffusion more closely resembles what we would see for polymer mixture without the presence of colloids, which we could essentially consider to be a phi of infinity. We can also see, especially at lower phi values, that the diffusion begins to increase, but the concentration of polymer ultimately dominates as the medium is heating up, so the diffusion begins to decrease. We can see this with our viscosity as well, which we learned from the Stokes-Einstein equation is inversely proportional to diffusion. So the viscosity begins to decrease, but then starts increasing as those polymers begin to aggregate. We can see that at a higher phi, the relative viscosity closely resembles our polymer-only medium. To summarize what we accomplished, we were able to track multiple particles in a medium heated under a microscope. We extracted the viscosity and diffusion of different media using the Stokes-Einstein equation and other equations we talked about. And we determined that polymer concentration had a dominating effect on the behavior of complex fluids as they are heated. Looking ahead, there's still much more work to be done. We have started to explore polymerized modes of droplets. So, our colloids are capable of undergoing a polymerization reaction to change their morphology, which could influence the behavior of the medium. We can also look into depletion interactions, so how the attractive forces influence the droplets when they get close to each other. Finally, we can investigate applications. Now, this complex fluid exhibits a thickening effect, where its viscosity increases with temperature. This could be very helpful for heating devices to have a much better temperature control. Thank you for listening, and thank you to everyone in the RIT Soft Matter Signaling Lab who helped me on my research this summer. I hope you enjoyed this presentation.